Thank you. I appreciate the opportunity to speak in this session, and I'm going to talk about biomarkers and other outcome measures, including patient-reported outcomes. And first of all, today we're talking about drug development, and particularly focusing on clinical trials. Why do we do clinical trials? We do clinical trials to isolate the pharmacologic effect of a product. What is its safety? What is its efficacy? And we want to distinguish the effect, pharmacologic effect of the product from spontaneous change. Many disorders go through cycles where they get better and worse, or maybe they just get better. And you want to distinguish when the patient gets better, is it because of the drug, or did they just get better? Sometimes in some disorders, there's a placebo effect. So if you give a placebo, the disorder gets better. You want to distinguish that from a true pharmacologic effect. And also, in any clinical trial, the study participant wants to get better. They want the drug to work. And the investigators want the drug to work. Even the regulators want the drug to work. So there's a potential for biased observation. So we do clinical trials to distinguish the pharmacologic effect of the product from biased observation, placebo effect, spontaneous change. We're going to talk about outcome measures because outcome measures are what we use to measure that effect. What you'd like to have in an outcome measure is something that's very sensitive. As the disease gets worse, as it gets better, as the clinical status changes, any small change, the biomarker can pick it up. That's a sensitive biomarker. You want something that's easy to measure and feasible. Many years ago, I was involved in a study where we were looking at muscle diseases, and we used to stick needles into muscles, and it hurt the study participants. So they never showed up for the clinic visits where they were going to have a needle exam. That's not feasible if the patients don't show up for the study. Want measures that are resistant to bias. Sometimes called objective, although objective is a little different than being resistant to bias. Reliable. If two different people do the measurement, do they get the same value? Something that's clinically meaningful, something that matters to patients. And at the end of the day, something that patients can understand. That is, we need outcome measures in our clinical trials that if a product is approved, patients have to sit down with their doctors and say, is this right for me? Well, if the outcome measure is something that patients and doctors can't understand, then it's very difficult for them to make that judgment about whether they want to take that drug. So we need outcome measures that patients can understand. Now, this ideal outcome measure, it does not exist. There is no ideal outcome measure. So we'll talk more about that. These are the types of outcomes. Clinical outcomes, and these measure, in general, how patients feel, function, or survive. And these include performance or functional outcomes, and we'll talk more about these, patient-reported outcomes, observer-reported outcomes, and clinician-reported outcomes. I'll talk a little bit more about these. In contrast to clinical outcomes, there are surrogate outcomes, which do not measure how people feel, function, survive. Rather, they predict or might predict how someone feels, functions, and survives. And in talking about these, we'll also talk about biomarkers. I want to talk first about patient-reported outcomes. These are measures based on a report that comes directly from the study participant about the patient's health. It includes symptoms or other things that only the patient could know. And sometimes it includes the patient perspective on things that someone else might observe. For example, if a patient is having difficulty walking, well, you could go to the clinic and the clinician could watch the patient walk and say they're having mild difficulty or moderate difficulty, so they could observe. But you can ask the patient too, how much difficulty are you having? A little difficulty? A lot of difficulty? Do you need a cane or a walker? So patient-reported outcomes can be things that only the patient knows, but they can also be things that the patient is judging their own ability to perform a task. Now, I want to emphasize that patient-reported outcomes are not something new. They've been around for hundreds of years. This is John Haygarth, and he was a physician in Great Britain back in 1799. And back then, they believed that metallic rods had healing powers. And if a patient with rheumatism, with joint pain, if you hit that patient with a metallic rod, that would alleviate the pain. Dr. Haygarth was a skeptic, and so he prepared wooden rods to look identical to metallic rods. This was a comparison to a control. He took 5 patients with joint pain, hit them first with the wooden rods, and then with metallic rods, and said, it just doesn't matter. And so he concluded that metallic rods were of no benefit, and the imagination can cure diseases of the body. Now, you can imagine if you've got joint pain and someone hits you with a rod and says, how is your pain? You might be motivated to say my pain is better so that they don't hit you with the rod again. The important concept from Haygarth was that this was a patient-reported outcome. And so it was important to conceal the treatment. So the wooden rods look just like the metallic rods. Now we use blinding or masking whenever feasible, and I want to emphasize that blinding or masking is essential for patient-reported outcomes. They're very difficult to interpret in the absence of blinding and masking. They're often difficult to interpret regardless. Other examples of patient-reported outcomes: pain intensity scale, the SF-12 Minnesota questionnaire. I'll say a few words about a couple of these. And sometimes patients can just report the number of events. How many headaches did you have over the past week? How many times did you vomit over the past month? So patients can report a number of events that only they would know. This is a pain scale that I made up, but it looks just like all the other pain scales. How severe is your pain? 0 is no pain. 10 is the worst pain imaginable. And just give it a number that matches your pain today or matches your pain over the past week. This is a patient-reported outcome, very simple and straightforward. The SF-12 is a much more complicated patient-reported outcome. It consists of 12 questions. Each question gets a score and the total score is added up to 0 to 100. And so you look to see, when patients answer the question, overall, did their score change by 2 points or 3 points or 6 points? But knowing what that 2 points or 3 points or 6 points means can be very difficult. So complicated patient-reported outcomes like this that combine many different questions can be difficult to interpret. I want to turn now from patient-reported outcomes to surrogate outcomes, including biomarkers. Biomarkers are defined characteristics that are objectively measured to indicate, is there a disease here, or what is the status of the disease? Is it getting better, getting worse? Look for the effect of an intervention. Again, these are not a measure of how a person feels, functions, and survives. And when we use the term biomarker, we mean there's not sufficient evidence that this is predictive of a clinical benefit. A surrogate outcome is a biomarker which also has evidence that it is predictive of a clinical benefit. So in characterizing surrogate outcomes, we're often trying to get data to support that there is a correlation between a biomarker and something that matters to patients. So a biomarker, a surrogate outcome substitutes for the endpoint of interest. These are some examples, some well-known examples of surrogate outcomes. People who have hypertension. What you're worried about if you have hypertension, high blood pressure, you're worried about death, stroke, heart attack. Those are the things that matter. But measuring those things takes a long time. So instead, we measure blood pressure. Well, you don't really care whether your blood pressure goes up or down so much. What you really care about is survival and heart attack and stroke. So we measure blood pressure as a surrogate to predict these other things. Similarly, in patients who have AIDS, we measure the viral load in the blood. In oncology, in cancer trials, we look at the size of the tumor. We think that, if the tumor gets bigger, you won't live as long. If the tumor gets smaller, you'll live longer. It's a predictor of clinically meaningful survival. What are the advantages of surrogate endpoints? We use surrogate endpoints because they are easier to measure, you can measure them earlier, and they're cheaper, and so clinical trials and drug development can be cheaper and faster, and we can get products on the market sooner. Disadvantages, sometimes they don't work. For example, we know that there are drugs that shrink the size of tumors in oncology and have no effect on survival. So the surrogate can overestimate or underestimate the true clinical effect. How do you find a biomarker? How do you find a biomarker and a surrogate? Well, talk to people. First of all, you want to understand the pathophysiology of the disease. Talk to the scientists who understand diseases. Look at the literature about that disease. Also, try to understand not just the pathophysiology of the disease, but the clinical course of the disease. And this can come from natural history studies. It can come from real-world evidence, which has medical records and information from patients, registries, clinical trials, and again, the published literature. Learn everything you can about the pathophysiology of the disease and the clinical course of the disease. And when you do this, you'll identify some laboratory measures, and that could be a chemical or a protein or a cell, like a white cell count or a platelet count. It could be an imaging finding. It could be maybe the liver gets bigger, or maybe the liver gets smaller, or maybe the brain shrinks in size, or maybe the brain swells. Imaging measure, a laboratory measure, something that can be objectively measured that will change as the patient's clinical status changes. In addition to patient-reported outcomes and biomarkers and surrogate endpoints, I want to mention performance or functional outcomes. These are very popular as outcome measures in clinical trials, and these are generally based on performance of a standardized task, often administered by a trained observer. Examples, a 6-minute walk test. You ask the patient to walk for 6 minutes and just measure how far they could walk and see if that changes as the disease changes. Word recall. Give the patient some number of words to remember and see how many they can remember. Performance functional outcomes are very common in clinical trials. Now, I want to give an example. This is purely hypothetical. I made this up. Hypothetical example, imagine mild Alzheimer's disease. And the first column here on the left shows those ideal characteristics that we talked about. Is it sensitive? Is it feasible? Is it resistant to bias? Is it clinically meaningful? The next three columns describe specific outcome measures that might be used in an Alzheimer's trial. First, memory. How well can you remember items? The next column, the SF-12, a patient-reported outcome. And next, measuring amyloid, measuring protein in the spinal fluid or in the brain, a biomarker or surrogate endpoint. And I've given each of these things a grade on the characteristics of an ideal endpoint. 3+, the strength of that outcome. 0, a weakness, and plus and 2 pluses in between. And as you can see, performance outcomes get 3 pluses for clinically meaningful and 3 pluses for patients' understanding. If patients understand, how many words can you recall? And it's clinically meaningful. But it can be subject to bias. SF-12, again, 3 pluses for clinically meaningful, but again, subject to bias. In contrast, the biomarker, amyloid protein, very sensitive. That's the strength here. Resistant to bias. So when it comes time to choose an outcome measure, don't think about what is the perfect outcome measure. There is no ideal outcome measure. Think about combining outcome measures in your clinical trial, combining outcome measures that complement each other. So if you're using a biomarker, well, that's fine. Use amyloid, that's fine. It's very sensitive and will detect small effects, but it's not clinically meaningful. So you want to have another outcome measure in that trial, such as a patient-reported outcome or a functional outcome, to complement. So you look at the strengths and the weaknesses of each outcome, and you have primary endpoints and secondary endpoints that complement each other. Sometimes people get too locked into what is the right outcome measure for this trial, and there's no perfect outcome measure. Interpreting a trial means understanding what happens to so many different aspects of the disease. Now, how do we use surrogate outcome measures? Well, they can be used for product approval. So the FDA has two types of approval. They have what we call traditional or regular approval and accelerated approval. And they're a little bit different. Both types of approval require well-controlled clinical trials. They both require substantial evidence of effectiveness. So the quality and the quantity of the evidence is just as much for accelerated approval as it is for regular approval. So sometimes people think, well, if I'm doing accelerated approval, I don't have to have as much data, or my data doesn't have to be as rigorous. No. Quantity and the quality of the evidence has to be just as good, and there needs to be enough safety evidence to support that there's a favorable benefit-risk balance. So what are the differences between accelerated approval and regular approval? First of all, the critical difference is the endpoint. The primary endpoint that supports approval for traditional approval is something that matters to patients, how patients feel, function, or survive. The primary endpoint that supports approval for accelerated approval is a surrogate endpoint, which is in and of itself is not clinically meaningful, but that predicts something meaningful. So the benefit under traditional approval is proven, well established, but the benefit under accelerated approval has not been proven. So if something has accelerated approval, it has no proven benefit. It has a benefit that is reasonably likely. Because the benefit is proven with traditional approval, there's no requirement for a confirmatory study. But with accelerated approval, because the benefit has not been proven, there is a requirement for a confirmatory study to prove that it truly works. Few references. The best glossary published by the FDA and the NIH defines biomarkers and clinical outcomes and surrogate endpoints and all the terms that are relevant here. A couple of guidances from the FDA, 2009 guidance on patient-reported outcomes, in 2024 a draft guidance on accelerated approval. I need to, I was at the FDA for almost 20 years and worked with so many wonderful people. I want to mention a few of those that helped me think about clinical trials and helped me prepare for talks like this. Dr. Kimberly Benton, who's now a regulatory consultant. She's left the FDA, but Drs. Rachel Anatol and Ann Rousey are still at the FDA. These are difficult times at the FDA, and I look forward to hopefully next year when they'll be back at this meeting. And that's my contact information, wilsonwbryan@outlook.com. If there are questions that you have that we don't have time for today, then I'll be happy to take questions. And I'm going to stop there. Are we taking questions?
Yes, thank you very much. Yes, we invite everyone to ask questions. We have 3 microphones, so please come and ask your questions. Maybe I can start by asking you a question. So thank you very much. It was a great presentation. So I wanted to ask a question about accelerated approval biomarkers. And maybe you can give us an example of when a biomarker was approved for accelerated approval process, but was not confirmed in a confirmatory studies later on. If possible, maybe neurologic space. Well,
I mentioned one, which was in oncology, that there have been tumor size has been used. In addition, folks have looked at in cardiovascular disease, people have looked at levels of low-density lipoprotein and other lipids. In some cases, those have not borne out as truly being predictive of mortality. So these, fortunately, the number of times when a biomarker has failed is the minority of times. When things get accelerated approval, usually the confirmatory studies confirm the benefit, not always.
Thank you.
Hey there, I'm Chrissy. I'm a parent advocacy group. And as we look and think about preparing for clinical trials, is there as a rare disease also, is there a minimum or ideal number that you need to have with the biomarkers?
So you mean number of biomarkers that you need to have for a clinical trial?
Yeah, what you're measuring against to be able to show that the clinical trial was a success with the drug that you're using. So is there a certain number that the FDA is expecting or makes it better or worse? I mean, you may have stronger ones and weaker ones, so do you put all of them in or 1 or 2? What's the...
Right. So what you're trying to do is choose outcome measures, including biomarkers, that fully characterize the disease. Some diseases are extremely complex. Pediatric neurodevelopmental diseases, very complex, affect physical function, affect cognitive function, and so you're likely going to need a lot of different outcome measures in order to characterize the disease. Some other outcome measures, some other diseases are much simpler in a way. Hypertension. Hypertension in many ways is a much simpler disease. I'm going to get in trouble here. And you can get away with just one surrogate endpoint of blood pressure in most cases. So the complexity of the disease is probably going to influence how many measures you need, including how many biomarkers. But it's going to be a case-by-case basis, looking at the disease and looking at what the biomarker does. And I want to also mention different stages of a disease may call for different biomarkers. You might find that early in the disease, one biomarker picks up the changes, and later in the disease, that biomarker isn't sensitive anymore, and you need a different biomarker. So it's not easy, and it's case by case for specific biomarkers and specific indications.
I have another question here on the left.
So does the confirmatory, I'm over here on your left, over here.
Oh,
Hello. Does the confirmatory trial have to be a separate trial, or can it be a continuation of sort of the original trial? I'm thinking more in an ultra-rare disease space. You do a data cut halfway, you get your accelerated approval, and just continue to follow those same patients, or do you have to start over?
So the question is, these rare diseases, you have to get as much information as possible from every single patient who participates. And so in some cases, it's acceptable to have the approval based on, say, 6 months of study or 1 year of study, and then the confirmation of the benefit comes with following for a longer period of time, 1 year or 2 years, maybe 5 years. So confirmation in some cases can be continuation of the study that supported approval.
Great, thanks.
You have time for one more question.
Thank you. Hi, my name is Olga. I'm from Myrtle. My question is it obligatory that if you have several, for example, several biomarkers as your several endpoints, that each one of them need to have a statistical correlation with your functional, clinical, significant outcome?
So the question is, if I'm using several different biomarkers, do they have to correlate with something that's clinically meaningful? Do they all have to correlate with something that's clinically meaningful?
And the question, is it like obligatory that your primary, like if you use one biomarkers, your primary endpoint, it needs to correlate statistically with you?
So to be a surrogate endpoint, it has to be reasonably likely to predict a benefit. And part of convincing the FDA and convincing other people that something is reasonably likely to benefit means having evidence that there is a correlation between changes in that biomarker and something that a patient feels, functions, and survives. Now, how strong does that correlation have to be? That's sort of the eye of the beholder. That's a judgment call. And you get that correlation data from registries, from real-world experience, from previous clinical trials, from the literature. But yes, if it's going to be a surrogate endpoint, the FDA has said, at the very least, there needs to be a correlation.
Thank you.
Thank you very much.
Yes, thank you, Dr. Bryan.